It's my pleasure to introduce Ozgur. He graduated from a veterinary school in Turkey in 2015 and completed his master's degree at Mississippi State University in 2020. He then began working at the Mediterranean Fisheries Research, Production, and Training Institute under the Ministry of Agriculture of Forestry in Turkey. In 2024, he started his PhD in the Fish Health and Disease Lab at Texas A&M University. Thanks for being here. Looking forward to your talk.
Hello, thank you for introduction, Nick. Hello, everyone. Today, I would like to talk about uh, of our research titled with "From a Catfish Pond to the Ocean: Adaptive Response of Aramus Hydrophila to High Salinity." This presentation will cover Aramus hydrophila and its impact on the United States catfish aquaculture with a focus on motile Aramus septicemia. I will discuss the role of salt in disease management, our study objectives, methods, key findings, and their implication. Finally, I will highlight practical applications and future research directions. Catfish aquaculture is a major United States industry valued at 435 million annually, according to USDA 2024, with key production in Texas, Mississippi, Alabama, and Arkansas. Farmed in earthen ponds, these systems create a complex environment where disease dynamics remain poorly understood. Investigating environmental factors like water quality management is crucial for improving disease control and sustaining production. Aromonas hydrophila is a gram-negative motile bacterium commonly found in freshwater environments. Typically, it's known as an opportunistic pathogen, meaning it usually causes secondary infection. But in 2009, an atypical strain of Aromonas hydrophila emerged as a primary pathogen in the United States, and significant disease outbreak known as a motile Aromonas septicemia. This has caused substantial economic losses with the millions of pounds of catfish affected, particularly impacting the Alabama. Motile Aromonas septicemia in catfish presents with a visible hemorrhages, ulcers, necrotic skin, and swollen abdomens. Other signs include exophthalmia, abscesses, gill hemorrhages. Aromonas hydrophila is causing septicemia with severe organ damage too. Why we want to do this research is because of this paper in Alabama. Our colleagues made the research. This publication in the research they identified the farm size key risk factors for Aromonas hydrophila outbreaks in catfish farms. According to their result, routine salt addition is not making any difference. But if they reach the optimal or make it above the 135 ppm of the chloride level, they saw that the outbreak decrease. This suggests that salt use alone might be ineffective unless optimal chloride levels are maintained. For that reason, we want to do this research, and our main objective of our study were to investigate how Aromonas hydrophila adapts to environments with varying salt concentration, and to understand how this adaptation affects its ability to cause disease. Understanding this adaptation is crucial because it reveals how environmental factors can influence microbial pathogenicity, specifically in aquaculture system, especially in earthen ponds. Ultimately, this knowledge helps us develop better strategies to prevent and manage disease outbreaks in farm-raised catfish. In this study, we explored the effects of different salt concentration on Aromonas hydrophila by exposing the bacterial culture to various levels of sodium chloride, which is 5 ppt to 50 ppt. All cultures were incubated at 28 degree. And before I go further, I would like to mention the average seawater is 35 ppt. So we go higher than the average seawater level of the salinity. We conducted the growth curve analysis to monitor bacterial growth under the salt conditions. Additionally, we performed the biofilm quantification assays and utilized both light microscopy and scanning electron microscopy to visually assess the morphological change induced by varying salinity. These methods allowed us to comprehensively analyze how salt concentration influenced the growth dynamics and structural characteristics of Aroma's hydrophylla. Now moving on to our result. When we analyze the growth curves of the Aroma's hydrophylla at different salt concentration, we observe the distinct patterns. At lower salt concentration, at the 0.5 and the 1% sodium chloride, the bacterium exhibited robust growth, reflecting that optimal or near optimal conditions. However, at higher salt concentration, especially at the 5%, which is 50 ppt of the salt concentration, bacterial growth was significantly reduced. And this indicates that higher salinity clearly exerts stress, limiting the bacterial growth. And we also want to check what happens if we remove the salt stress from the culture. For that reason, we have done the reverse passaging. When we reverse passage it from the 5% to regular media, 0.5 salt concentrated media, we didn't see any differences. So that means high salt levels are not causing any permanent changes in a short term. As for the biofilm quantification, this graph illustrates the biofilm production of Aroma's hydrophylla under different salt concentration. Lower salt concentration showed increased biofilm formation. In contrast, at higher salt concentration, 2%, especially at 5%, we didn't observe any biofilm formation or reduced biofilm observation only. This result indicates the salt is inhibiting the biofilm development. And we also want to check the bacterial morphology under the light microscope. For that reason, we grow our bacteria at different salt concentration, and at the lowest salt concentration, which is 5 ppt, the bacteria appeared well adapted, showing typical rod-shaped morphology, normal cell division, and dense population. And that is indicative of the active growth. And when we increase the salt concentration at 1%, we start seeing visible morphological adaptation, but it was not too much. But when we go further to the 2% of sodium chloride, we observe morphological change in Aroma's hydrophylla. Under the microscope, bacterial cells appeared noticeably elongated and filamentous, and it's a clear indication of the significant stress responses. It's a clear indication of significant stress responses. When we go further to 5%, Aroma's hydrophylla showed dramatic morphological alterations under the microscope. We observed extensive filamentation, highly elongated, irregularly shaped cells. Additionally, bacterial density was severely reduced. We want to take a closer look with a scanning electron microscope. For that reason, we use the same culture media with the same bacterium, and then we look at under the scanning electron microscope. And for the 0.5 sodium chloride, the bacterium maintained typical rod-shaped morphology. The cells appeared to be dense, supporting our previous observation from both light microscopy and biofilm quantification assay. When we increase the salt concentration to 1% sodium chloride, it shows that early signs of the morphological stress responses, but it was not too much like the light microscope result. When we increase more to the 2% of sodium chloride, SEM revealed the significant morphological alterations in Aroma's hydrophylla. The bacterial cells exhibited elongation, with some showing filamentous structures. When we go further to 5% of sodium chloride, Aroma's hydrophylla exhibited extreme morphological stress under scanning electron microscope. The majority of cells appeared highly elongated, filamentous, and structurally deformed. We also want to check the biofilm formation under the scanning electron microscopy. At lower salt concentration, bacteria was forming the biofilm normally as we expected from the bacterial biofilm formation curve. But when we increase the salt concentration, the bacteria started showing reduced biofilm formation, and we start seeing the elongated cells too. To further investigate the effects of the salt stress on the aromatase hydrophila, we examined the bacterial motility under different salt concentration. Motility plays a crucial role in bacterial virulence. Our results showed that at lower salt concentration, particularly in 0.5 and 1%, motility remained high, allowing the bacteria to spread efficiently. However, at higher salt concentration, especially at 5%, motility was almost entirely absent. This finding suggests us the elevated salt levels hindering the motility. We also wanted to check the susceptibility of aromatase hydrophila against the oxytetracycline in different salt concentration. The bacteria grown in high salt concentration were highly susceptible to oxytetracycline comparing with the low level of salt. To ensure the accuracy of our experimental results, we performed the molecular confirmation of aromatase hydrophila across different salt conditions. Using PCR-based analysis, we confirmed the presence of the bacterial DNA in all test conditions, and all of them are the same. Our key observation and findings: when we increase the salt concentration, the bacterial growth is decreasing. And the increased salt concentration is changing the cell size and cell shape and decreases the biofilm formation too. In conclusion, our study demonstrates that aromatase hydrophila can adapt to varying salt concentration, but its growth, morphology, biofilm formation, and motility are significantly impacted by salinity level. These findings emphasize the complex role of salt in bacterial pathogenicity. For the future directions, we want to investigate the long-term effect of the salinity on bacterial resistance mechanism and develop targeted mitigation strategies for aquaculture system, and explore the pathogenicity and immunogenicity of aromatase hydrophila in different salt concentration, and explore the interaction between the aromatase hydrophila and other aquatic pathogens in the high salinity environments. I would like to acknowledge the USDA NIFA who support my research, and I would like to thank my department and my college too. Thank you for your attention, and I can take questions.
Thank you very much, Ozgur. Folks, if you've got a question, feel free to unmute yourself and ask it, or drop it into the chat. We've got time for a quick one. Oh, job well done. No questions. Thank you very much.
Okay, thank you.